Hello, everyone. My name is Young Lee, and I'm the president and CEO of the Damon Runyon Cancer Research Foundation, as well as a proud former Damon Runyon Fellow. Damon Runyon's mission is simple: funding brave and bold. We provide funding to the most brilliant young scientists to enable them to pursue high-risk, high-reward research that will enable breakthroughs in the understanding and treatment of all cancers. We have a long track record of success, and over the past 75 years, we have invested over $430 million in over 4,000 scientists. I'm very pleased to welcome you today to our first Damon Runyon Catalyst webinar. The goal of this webinar series is to highlight the career paths and personal journeys of scientists across all areas of cancer research. Today, we are delighted to be hearing from William G. Kaelin, Jr., MD. Dr. Bill Kaelin is the Sidney Farber Professor of Medicine at the Dana-Farber Cancer Institute and Harvard Medical School, and a Howard Hughes Medical Institute investigator. He has numerous scientific accolades, too many to list here, which you can read about in his biography online. Most notably, he was awarded the 2019 Nobel Prize in Physiology or Medicine for his work on the HIF class of transcription factors, which are important for how cells sense oxygen. His work has paved the way for promising new strategies to fight cancer and many other diseases. He will tell you more about this work today. Throughout his distinguished career, Bill has championed the importance of investing in scientists in the vulnerable early stages of their careers. We are very fortunate to have had Bill's involvement in the leadership of Damon Runyon for over 15 years. Bill is a member of Damon Runyon's board of directors and was elected vice chair of scientific programs. This esteemed group of scientists sets the priorities and strategies for the foundation. He currently also serves as chair of the Physician Scientist Training Award Committee. And as a physician scientist who took a later turn into cancer research, he has taken special interest in increasing the ranks of physician scientists and mentoring the next generation of leaders in all parts of cancer research. We are honored to host Dr. Kaelin today to hear him share and reflect on his own personal and professional journey with his talk titled, How I Accidentally Became a Scientist and Won the Nobel Prize, and Why Science is Like Fishing. So with this, I'm going to turn the Zoom over to Bill. And after Bill's presentation, we will have a Q&A session with an opportunity for guests to ask questions of Bill. And this will be moderated by Dr. Angela Brooks. Bill,
Great.
I'm going to turn it over to you. Thank you.
Thank you so much, Young, and thank you all of you who are sharing some time with us online. And I'll see if I can share my screen and hopefully turn off closed captioning and hopefully get a pointer, and then we'll be all set, I think. Very good. So thank you, Young. So I was actually born in Queens, New York, and grew up in the suburbs of New York, first on Long Island and then in Fairfield, Connecticut. You know, looking back at my childhood, I would say I had devoted parents, but they weren't what some people might refer to as helicopter parents. They weren't constantly hovering over me. I had some great teachers. I had ample unstructured time where I was just told to go play and amuse myself. And I think that's important. I think a lot of scientists will tell you that growing up, they had some unstructured time to get into some mischief now and then. And I had a lot of toys that fostered an interest in science and engineering. I had a microscope, a chemistry set, construction toys, electric race cars, etc. For example, here's my toy microscope that I think my mother gave me when I was about 8 or 9 years old. Now, frankly, it wasn't unusual for boys in the 60s to have such toys, and sadly, it was mostly boys back in the 60s. But the reason we often had such toys was the 60s was the time of the space race and the Cold War. So science and engineering were really celebrated and encouraged in the United States. And of course, what I'm showing you here is the Sputnik launch in 1957. And parenthetically, I was born in 1957, which really set in motion the space race and which heightened the Cold War. Now, if I was to describe myself as a young student in elementary school and even in high school, I would say that I liked learning concepts and ideas. I liked objective subjects where there was a right or wrong answer. And like a lot of young people, I liked playing. I strongly disliked memorizing facts. I hated rote memorization. I didn't particularly like subjects that I viewed as being very subjective. And I'm sad to say, I didn't particularly like studying. So as a result, in high school, for the most part, I was a somewhat mediocre student. I got mostly B's. Fortunately, the one thing that saved me was I must have gotten my mother's math gene, so to speak. She had been a mathematician when she was younger. And in fact, she was an actuary at Metropolitan Life Insurance Company before she began raising a family of 5 children with me as the oldest. So math always came easily to me, and I could get A's without studying, which meant, so in other words, this was my kind of course. And so my typical transcript when I was in high school would be, A's in math, and then more or less B's, and maybe even the occasional C and everything else. But then something changed my life. I was in a public high school in Fairfield, Connecticut, and my high school was one of the first high schools in the country to install a computer terminal. Now, mind you, this was long before the invention of the personal computer. So this computer terminal, which was basically a glorified IBM typewriter, was connected to a mainframe computer in a nearby university, Fairfield University. And I became interested in computer science, this new field of computer science, because to me, this was sort of mathematics come to life. And I was also just fascinated with this idea that you could send a command to this big, powerful machine and it would have to respond to you. And then one day in the waste paper basket next to this computer terminal, I saw a pamphlet inviting high school students to apply to do a summer program to study college level mathematics and computer science at Florida Atlantic University. And somewhat on a whim, I applied and was accepted. And so off I went to Florida Atlantic University, and I spent 8 weeks with 31 other high school students studying college level math and college level computer science. Now, I have to tell you, I was sort of scared to death doing this because I assumed every one of these students would be a genius, and I certainly wasn't a genius. But I was sort of pleasantly surprised to find out I wasn't the dumbest of the 32 students. In fact, I could more or less hold my own with most of them. But what they all had in common was they all had much better grades back home in their respective schools because, of course, all of them had much better study habits than I did. And I also came to appreciate that one of my problems in school, in hindsight, was that I was simply bored because I wasn't being sufficiently challenged, because I really enjoyed my 8 weeks down at Florida Atlantic University. And so one of the very important things I learned from this experience was you grow the fastest when you're surrounded by people who are smarter and more talented than you are. It might make you feel good if you're always the smartest person in the room, but I guarantee you, you'll grow faster if you can surround yourself by terrific people who are very talented and very smart. And so at every turn, I've tried to do that throughout my career, because the people around you will make you better if you can do that. So I went back for my senior year and I decided to do an experiment. I decided that, maybe for at least 1 year in high school, instead of sitting in the back of the classroom and making fun of the smart kids, I would sit in front of the classroom and actually try to be engaged and try to be one of the smart kids. I decided I would actually bring my books home and actually do my homework each night, which was a new concept for me. And I actually decided I would sign up for what I thought were the most challenging courses. And so this experiment thankfully worked. I was now an A student rather than being a mediocre student. So this was really quite life-changing. And I then was fortunate enough to get into Duke University, and I decided I would be a pre-medical student. Now you might say, but Bill, what happened to mathematics and computer science? And I'm embarrassed to say I just wasn't smart enough to see the personal computer revolution that was still about 10 years or so away. And my idea of what a career in computer science would be like at that time was that I'd be sitting in a windowless room writing computer code for military and industrial applications, and that didn't seem very exciting to me. And I thought, at least by going into medicine, I could use mathematics and science to do some good in the world. And I also could interact with people on a daily basis. But if I'm really being honest, I think I was also influenced by certain TV shows, such as Medical Center, which was popular back in the 70s, starring Chad Everett, where a typical episode would be that the dashing young surgeon would do some intricate life-saving surgery, and then would leave the hospital, jump in a red convertible, and go play tennis with a beautiful young lady. And so, as a high school person, college person, sign me up. That sounded pretty good. So I decided I'd try to go to medical school. Now, once I got to Duke, I was actually doing pretty well. I decided to major in mathematics and chemistry. I mean, you can figure out why I majored in mathematics. I majored in chemistry because I needed to take so many chemistry courses to be a pre-med that, I only needed to take one or two more to fulfill the requirements to be a chemistry major. Parenthetically, I took as few biology courses as I possibly could because I thought biology was sort of boring and descriptive at that time, and certainly involved a fair amount of rote memorization. Now, when I was a junior at Duke, one of my fellow students told me, if you're going to apply to medical school, it would look really good on your application if you had worked in a laboratory, sort of rounding out your transcript. So I met with a Duke chemistry professor who told me he had a project that 7 previous undergraduates had worked on, and all of them had gotten into medical school. So of course I was very happy because I thought I had found the golden ticket to medical school. So I immediately signed up to work in this lab. However, if I was a little smarter and a little wiser, I might have asked, why couldn't these 7 previous people bring this project to completion? And I became the 8th person who can bring this project to completion. And I can now tell you with absolute certainty that this first project I was given in college was uninteresting, unimportant, and undoable. And so I simply floundered aimlessly for 18 months. Because I did flounder so terribly, my professor rewarded me with a C-, which I can tell you for a pre-medical student is sort of like having a wooden stake driven through your heart. And then as an added sort of punitive flourish, he wrote in the margin of my transcript, "Mr. Kaelin appears to be a bright young man whose future lies outside the laboratory," which is a little bit like being told you have a good face for radio. Now, despite this blemish, I did eventually get into medical school. And I also learned another very important lesson. And that is, if you're struggling in a laboratory, it might be your fault, but it could also be that you're simply in the wrong lab, or you have the wrong project, or you have the wrong mentor. Now, ironically, this C- did have its intended effect on many of my medical school applications. So I was rejected from Harvard Medical School, where I'm currently a professor, as you heard. But I did allude to the fact that I did fortunately get into medical school. I did wind up going to Duke Medical School. So this is me circa 1979. Now, it turned out that one wonderful thing about the Duke Medical School curriculum was in your 3rd year, you were encouraged to do a laboratory research project. So with considerable trepidation, I did work in a laboratory during my 3rd year of medical school studying tumor blood flow. Here I am at my first scientific poster. I can tell you it was a little bit lonely. I think only 3 or 4 people came by to see my poster. My mentor was a radiation biologist named Randy Jertle. Here's actually my first paper describing the effect of a drug called verapamil on tumor blood flow. But one of the things I learned from Randy is when a young person is entering the laboratory, it's really important that within a reasonable time frame, they have the taste of at least a little success, so they can start to experience the joy and the excitement of working in the laboratory. And so thankfully, I was more successful in Randy's lab than I was in my undergraduate laboratory, but this wasn't enough to convince me that I could be a scientist or even wanted to be a scientist. I really thought I wanted to be a clinical doctor at this point. I was fortunate enough to go to Johns Hopkins to train in internal medicine. Here I am as an intern in 1983. And I was so sure I wanted to be a clinical doctor that I actually spent an extra year at Johns Hopkins as one of the chief medical residents. And so that's sort of where I stood in 1987. I became interested in cancer medicine, and I was fortunate enough to be accepted to do a fellowship at the Dana-Farber Cancer Institute to study medical oncology, or cancer medicine, if you will. But then I had the very, very good fortune of meeting the late, great Dr. David Livingston, who for many years was also on the Damon Runyon board. So as part of my fellowship training, I was again given the opportunity to work in a laboratory, and just through sheer dumb luck, my mentor turned out to be David Livingston. And David's really the one who taught me how to do cancer research, how to do cancer science. And one take-home message I want to emphasize is much of learning how to be a scientist is done by apprenticeship. And mentorship really, really matters. And I was just so fortunate to have David Livingston teach me how to do science, how to think like a scientist, how to design penetrating experiments, and how to interpret data, form hypotheses, etc. So I had some success in David's lab, and I started my own laboratory in 1992. And one of the most important things a young scientist has to do as an independent investigator is decide what problem or problems am I going to work on. And fortunately for me, shortly after I started my laboratory, this paper appeared in the scientific literature, which described the isolation of a gene that, when mutated or altered, caused a hereditary cancer syndrome that doctors refer to as von Hippel-Lindau disease. And fortunately, because of my clinical training, I knew a fair amount about von Hippel-Lindau disease. So here I'm showing you various kindreds of various families with VHL disease, and every filled circle or square is a individual in that family who's developed cancer. And the cancers that you typically see in these families include kidney cancer, a blood vessel tumor called hemangioblastoma, and an adrenal gland tumor called pheochromocytoma. And so these individuals develop cancers typically early in life. They can be in their 20s, 30s, and 40s. And so clearly there was a problem in these families. And as I said, this other group had now isolated the gene that, when altered, was responsible for this high risk of cancer generation after generation in these families. Now, I also knew from my clinical training that kidney cancer was one of the 10 most common cancers, and I thought, everything else being equal, why not work on a common cancer as opposed to an uncommon cancer? And it was pretty clear from these unfortunate families that the VHL gene somehow likely played a pivotal role in the development of kidney cancer. Now, just to bring you all up to speed, individuals with von Hippel-Lindau disease have inherited a defective version of the VHL gene from mom or dad. So in this schematic, it's the version from mom, the so-called maternal allele, that's defective. But initially they're okay because the other copy, the copy they got from the other parent, is intact. But unfortunately, if you're born with a defective version of the VHL gene, you have about a 90% chance that at least one cell in your body will spontaneously lose the remaining copy of the gene, and it's that cell that can go on to form a tumor. Now, as you would predict from the knowledge that inheriting a defective VHL gene from mom or dad predisposes you to kidney cancer, if you now look at non-hereditary kidney cancers, so garden variety kidney cancer, you again often see that both copies of the VHL gene, the one from mom and the one from dad, are defective, mutated, if you will, or completely lost. But here, both the maternal and the paternal allele or copy was mutated sometime after conception. So the difference here being that in von Hippel-Lindau disease, you've already started out in life with a defective copy of the VHL gene. In most kidney cancers, these mutations occur after conception. Now, again, I knew from my clinical training that the tumors seen in von Hippel-Lindau disease are notoriously good at inducing new blood vessels to form to provide them with blood. Scientists refer to that as angiogenesis. And we knew that's because they frequently overproduce a hormone called vascular endothelial growth factor, or VEGF for short, which acts like a fertilizer for new blood vessels. And another curiosity is they sometimes cause patients to make too many red blood cells. And that's because these tumors sometimes produce a hormone called erythropoietin, or EPO for short, that stimulates the bone marrow to make more red blood cells. And I knew again from my clinical training that VEGF and erythropoietin are normally induced when cells or tissues are not getting enough oxygen. So it was almost as though the oxygen sensing mechanism in these tumors was defective or was somehow being hijacked. So the big aha moment was maybe the VHL gene plays an important role in oxygen sensing. Now, I have to just introduce a little bit of jargon for the non-scientists on the call. Scientists often refer to genes being on or off, or sometimes they'll say genes being expressed or not expressed. What they really mean when they say a gene is on or being expressed is they mean that the DNA of the gene is being copied into so-called messenger RNA, which is usually the blueprint for making a protein. And in general, the more mRNA you have for a given gene, the more of the protein you will have for that gene. And this is often under the control of a so-called transcription factor, which will turn that gene on. So that's what I mean when I talk about a gene being on or a gene being off. Now, we did a simple experiment, but for the young people, simple in science is not a bad thing. Simple is a good thing. You should always try to do the simplest experiment that clearly answers your question. So we did a simple experiment in the laboratory using standard tools. We created cells we could grow on a plastic dish where the VHL protein was intact or the VHL protein was absent or defective. And we grew those cells in chambers that had low oxygen or high oxygen. And then we measured the abundance of many mRNAs that are normally induced by low oxygen, such as that mRNA for vascular endothelial growth factor. And the technical term for low oxygen is hypoxia. So as expected, if the VHL protein was present, these so-called hypoxia-inducible mRNAs, such as the mRNA for VEGF, only accumulated under low oxygen conditions, whereas if the VHL protein was absent or defective, these cells lost the ability to sense oxygen, and they produced high levels of these mRNAs, whether oxygen was present or not. So our hypothesis was correct that the VHL protein was somehow required for sensing oxygen. Now, we knew from the work of multiple laboratories, including the work of my fellow laureates, Greg Semenza and Peter Ratcliffe, that many of these so-called hypoxia-inducible genes, such as the vascular endothelial growth factor gene and the erythropoietin gene, are under the control of a transcription factor that scientists refer to as hypoxia-inducible factor, or HIF. And HIF is made up of an unstable alpha subunit that's normally destroyed or degraded when oxygen is present, as well as a stable beta subunit. And so based on this clue, our lab and others showed that the VHL protein, together with these other proteins shown here, when oxygen is present, adds a little chemical tag to HIF alpha, which scientists refer to as a polyubiquitin chain. But for you and me, all we need to know is this serves as a signal for HIF alpha to be destroyed by a cellular garbage disposal we refer to as the proteasome. Whereas when oxygen levels are low, or the VHL protein is defective, such as in kidney cancer, now HIF alpha doesn't get destroyed. It can bind to that HIF beta protein and turn on genes such as VEGF and erythropoietin. So we had solved the mystery of why cells lacking the VHL protein overproduce things like VEGF and erythropoietin. But this begged the question, how does the VHL protein know, if you will, whether oxygen is or is not available, and hence whether it should or should not mark HIF alpha for destruction? And so it turned out our laboratory and my fellow laureate, so Peter Ratcliffe's laboratory, working independently and in parallel, showed that the signal goes like this. So in the presence of oxygen, there's a little chemical modification called hydroxylation of a amino acid called proline. So you may know that proteins are made up of 20 amino acid building blocks, one of which is called proline, which we represent with the letter P. And when oxygen is present, this little chemical modification takes place, and that serves as the signal for the VHL protein to bind. Now, this is exquisitely oxygen dependent. We know that the enzymes that do the work here, and enzymes are simply proteins that accelerate chemical reactions. So the enzymes that do the work here, which are variably called the EGLN or PHD prolohydroxylases, they look for molecular oxygen, and if oxygen is present, they use the oxygen to make that little chemical modification, that hydroxylation. And so this was a very satisfying link between oxygen availability and this chemical modification that could now dictate whether HIF alpha would be unstable or stable. Parenthetically, we quickly figured out that these enzymes also require not only iron, but they also require a chemical that scientists refer to as 2-oxyglutarate. And fairly quickly, pharmaceutical companies figured out that you could make chemicals or drugs that would prevent the utilization of 2-oxyglutarate by these enzymes, basically tricking the body into thinking it wasn't getting enough oxygen, stabilizing HIF and causing the body to make increased red blood cells, which you might imagine would be good if, for example, you were trying to treat anemia. And so, for example, there are a number of such drugs that have now entered clinical trials, one of which is called roxadustat. And I should confess I have a small financial conflict of interest in roxadustat. But roxadustat has now been approved for the treatment of anemia in a number of countries around the world, including in China. Here I am in Beijing in 2018, watching this drug being manufactured for the Chinese population. Now, returning to the role of VHL and HIF in cancer such as kidney cancer, our laboratory and others showed that in kidney cancer, there's a very specific HIF that's the problem. So when I refer to HIF, it's really a family of HIF genes, the best studied of which is called HIF1 alpha, and the less studied member of the family is called HIF2 alpha. And we came to understand that in kidney cancer, if anything, the problem is really HIF2 alpha. And in fact, in some kidney cancers, they get rid of HIF1 alpha altogether because HIF1 alpha has the opposite effect. HIF1 alpha tends to prevent the development of kidney cancers. So in kidney cancers that lack the VHL protein, they really now hijacked HIF2 alpha, and HIF2 alpha is the bad guy. So what can we do about this? Well, I showed you this slide earlier. We knew that HIF regulated a number of genes that we suspected played a role in cancers in general and kidney cancers in particular, such as vascular endothelial growth factor. And fortunately, by the 90s, a number of drug companies were developing VEGF inhibitors for the treatment of cancer. And we argued if these drugs were going to work in any cancer, they should work best in kidney cancer because of this fairly direct link between loss of the VHL protein, accumulation of HIF, and therefore the accumulation of too much VEGF. And I'm happy to report that I think we're up to 8 VEGF inhibitors that are now approved for the treatment of kidney cancer. And for some kidney cancer patients, these drugs are extremely effective. I know of some kidney cancer patients who have been on these drugs for years. So that's the good news. But the bad news is not every kidney cancer patient who's treated with a VEGF inhibitor will respond. And even those that do will eventually progress. So we have to figure out a way to do better. Well, I think you would argue based on first principles that maybe rather than targeting any one gene that's under the control of HIF, maybe we should target HIF itself. And based on what I told you, maybe we should target specifically HIF2 alpha, but or HIF2. But the conventional wisdom for many years was that HIF was going to be undruggable, that it just lacked the right sorts of nooks and crannies to be accessed with a small molecule drug. But fortunately, 2 wonderful scientists, Rick Bruick and Kevin Gardner, who were then at the University of Texas Southwestern, identified a potentially druggable pocket in the so-called PAS-B domain of HIF2 alpha, which is what I'm showing you here. And they identified chemicals that could bind to this pocket and in so doing induce HIF2 alpha to fold improperly such that it couldn't bind to DNA anymore. And these chemicals were then later out licensed to a biotech company called Peloton Therapeutics that was recently acquired by Merck Pharmaceuticals. And I, again, should declare I have a small financial conflict of interest. But Peloton shared with us one of their early HIF2 alpha inhibitors that they called PT 2399. And we could show in our laboratory models that this HIF2 alpha inhibitor did everything we would have hoped for. It decreased HIF-dependent mRNAs, such as the VEGF mRNA, and it decreased the ability of kidney cancer cells to proliferate both on plastic dishes and also when grown in mice. Now, eventually, an improved version of this drug called belzutifan went into so-called phase 2 testing in patients with advanced kidney cancer who had failed standard of care agents, including the VEGF inhibitors. So these are what doctors refer to as swimmer's plots. So each of these horizontal bars is a patient on the clinical trial and how long they were on the trial at the time of this analysis. So for orientation, here's 1 year on therapy. All the patients with the black arrows were doing well on the therapy still at the time of this analysis. And all the patients with the yellow stars had significant shrinkage of their tumors. And so based on these promising data, this drug has now advanced to phase 3 testing. But I mentioned these patients had failed multiple lines of therapy. They had failed VEGF inhibitors. They had often failed so-called immune checkpoint inhibitors. And a truism in medical oncology is that most drugs work better in sort of a frontline setting than when you reserve them for patients who have already received multiple other therapies. Unfortunately, we were able to convince Peloton and then Merck to treat a series of 61 patients with von Hippel-Lindau disease who had measurable kidney tumors that had never been treated medically. So these patients, in part because they can develop so many tumors, were in careful surveillance programs where they would get an MRI or a CT every 3 to 4 months. And only if their tumor changed its behavior or started growing beyond a certain size would they undergo surgery. And this wasn't an attempt to delay or prevent the need for repeated surgeries, which would otherwise leave them without a functional kidney. So again, this is sort of an experiment that begins to tell us what this drug might do in a frontline setting. So once again, these are so-called swimmer's plots. Here's 1 year on therapy. Now I think you can see that almost all the patients are doing well for a very significant period of time on the therapy. And although it's hard to see, these triangles and orange circles are showing you patients who had very significant tumor shrinkages. And in fact, in terms of tumor shrinkages, another way to look at the data are in these so-called waterfall plots, where each of these vertical bars is a tumor in a different patient. And I'm showing you the amount of tumor shrinkage in these tumors. And so anytime the bar goes below 0, there was measurable tumor shrinkage in these patients on this trial. Now, cancer statistics are okay, but I must say there's always an unfortunate thing that happens, which is you forget to remember that each of these bars or stars or dots is another patient. So just to put a human face on this, here's a patient on the VHL trial who was sharing on their social media page, even while she was on the trial, that she was responding. You can imagine this individual had seen this disease ravage their family generation after generation, and she had just assumed she would die sometime in midlife. And here she's saying, I never thought I'd see this day. And she's describing that some of her tumors were getting smaller or disappearing. And I should point out that in addition to the kidney tumors shrinking, in many cases, the other tumors these patients had, such as some of their brain tumors and adrenal gland tumors, also shrank or disappeared. And because everyone likes the video, here's a short video from another VHL patient on the trial.
Hey everybody, it's Justin. I just wanted to give you a quick update. I am in a gondola right now in Taiwan. Over there is Taipei 101. The gondola is actually right by the Taipei Zoo. But I just wanted to give you a quick update and say I'm doing well. I'm enjoying my trip. If it wasn't for the PT2977 drug trial, I would have never been able to come out here and do what I'm doing right now. So I just want to thank Peloton, and I hope Merck will fast track this drug for a VHL treatment. So if you guys are listening, hopefully you guys will put it on the market to help VHL. But yeah, keep watching these videos. I'll be making more, and I'll get better at it. And I have to get the angles right because it kind of looks fat, you know.
I like that video because at that age, that's the kind of thing you should be worried about, namely whether you look fat on your vlog. You shouldn't be worried about whether your next MRI is going to look so bad that your doctor tells you to get your affairs in order. And I'm happy to report that the FDA, in fact, did approve this drug for the treatment of von Hippel-Lindau disease last summer, and we're waiting for the results of the phase 3 trial in sporadic or non-hereditary kidney cancer. So in closing, why is science like fishing? Well, the first thing is in both science and fishing, you have to first start by learning some techniques, but it turns out that's one of the easiest parts of science. If you have the right person showing you how to do the procedure, you can usually learn how to do the procedure. So, you know, in fishing, it might be learning how to do a cast or bait the hook. In science, there are techniques to learn, but if the right person is showing you the technique, I guarantee you can learn the technique. But just as it's important to pick a good place to go fishing, it's important to pick a good problem to work on. And I would argue if I got one thing right, working on the von Hippel-Lindau tumor suppressor gene was a very good thing to work on. And like fishing, there's certainly some luck involved in this business, and every scientist knows this. Now, it's true, the more experiments you do, the greater your chances that over time, you'll make a nice discovery or catch a big fish, but there's certainly some luck involved. And frankly, if when we had arrived at the answer to how cells sensed oxygen, the answer that nature had arrived at struck people as being inelegant or clunky, or was a variation of something we had seen many times before in other contexts, there would have been no discussion of winning the Nobel Prize. But the minute we had the privilege of seeing the result, we were all pretty awestruck at how beautiful nature is and how wonderful this mechanism that nature had arrived at for oxygen sensing. And the final thing I'll say is when you do go to Stockholm, if you're lucky enough to go to Stockholm, one of the events is you go to the Nobel Museum and you donate one or two things of significance to you. And so when I went to the Nobel Museum, I did donate that microscope that my mother had given me as a child, as well as my medical equipment from when I was a health officer at Johns Hopkins. So with that, I will thank you very much for your attention, and I look forward to the Q&A.
Bill, thank you so much for sharing with us. It's fascinating to hear about your both your personal as well as your professional career. So at this time, I'm delighted to be able to introduce our moderator for the Q&A session, Dr. Angela Brooks. Angela is Associate Professor of Biomolecular Engineering at the University of California, Santa Cruz. And Angela was a Damon Runyon Fellow, actually in Boston, and a recipient of the Dale Fry Award for Breakthrough Scientists. Her lab works on developing computational and experimental approaches to study cancer mutations that cause changes in the transcriptome, particularly through RNA splicing. And at this time, I also want to remind all of our audience that you can please type your questions into the Q&A box in the chat, the Q&A box at the bottom of your screen, and we will ask as many questions as time allows. This webinar will conclude no later than 10 minutes after the hour. So with that, I'll turn it over to you, Angela. Thank you.
Thank you, Young. And thank you, Bill. That was a really great talk, and actually it was really interesting to hear your path. I heard some similarities, like I hated rote memorization as well. So yeah, so I'm going to start off with a few questions, but again, you know, if you have other, the audience, if you have other questions, please put in the Q&A and we'll try to get to them. So the first question is based on your fishing kind of analogy, and so what's your process for picking a scientific problem to work on? You know, thinking about how do you find the right place to fish? You know, do you reflect individually on these questions or discuss with colleagues?
Yeah, great question. So I always look back when I was a very young scientist getting started, and before that, even a student in college. And I think about how many things I got wrong about what a career in science would be like. So I already alluded to one. I thought learning the techniques would be one of the hard parts, but I've already told you, I'm sure you agree, if the right person is teaching you the techniques, you can probably learn the techniques. Then the second thing I got wrong was I thought you had to be a genius. And my life might have been easier if I was a genius, but I'm not a genius. I think I'm reasonably smart, but I'm careful and I'm curious. And those are the three ingredients I think you need to be a successful scientist. But now more to your question, I also thought science meant you stared at a blackboard and these great ideas just came out of nowhere and you had a bolt of inspiration and you knew immediately that's what you should work on. But I can see you smiling because you know that's not how that works either. I think what really happens is, first of all, hopefully you have a broad sense of what area you might want to work on. So I knew I wanted to work in cancer. Then the trick is, and this is important, I think the trick is to find something within that area that you find is interesting. And of course, what makes the world go around is different people find different things interesting. But going back to my uninteresting, unimportant, and undoable, the things I look for, you need to find a problem that's at least to you interesting. It's nice if you could imagine a way it could turn out to be important. And you need to have at least some imagined path forward where it can become doable. And this again is where mentorship is so important. And when I got started, like most young people getting started, my mentor played a profound influence in terms of helping me with the first questions I would work on, which brings me to the last point, which is once you get started and you're immersed in something and you start reading and you go to seminars and you talk to other scientists, the problem isn't finding a question to work on. The problem is how to prioritize amongst the many questions you actually could work on. Once you're in a field and you start to get some data and you start to learn more about more and more, that's the trick. How do I prioritize? And so for me, the way I prioritize was I relied on the power of genetics. So the definitive experiment, which unfortunately is tragic for the poor families that carry this defective gene, but the definitive experiment had been done. There was no question that the VHL gene played a role in kidney cancer. And so it wasn't a big leap to think if I learned about the VHL gene, I'd learned about kidney cancer. But then I also alluded to the fact I thought it was reasonable to assume you might also along the way learn about oxygen sensing. And again, oxygen sensing seemed like an important problem given so many diseases involve oxygen.
Great. Thank you. The next question is, what advice can you give trainees or students who are on the fence about remaining in science because of maybe economic or other reasons?
Yeah, another really important question. I think we in the scientific community, unfortunately, collectively do a good job of conveying to young people sort of some of the things maybe we don't like about science, some of the nuisances, gee, I had to write another grant, or my paper got rejected, or my experiment failed. So I think we do a very good job conveying the things we don't like about our jobs. We do an absolutely terrible job conveying all the things that are wonderful about our jobs. I mean, I think one of the greatest joys a person can have in life, and I'm not the first ever to say this, is to do a job you love so much that you would do it even if you didn't need to be paid. And so, I think we need to do a much better job conveying that, most people go to work each day because they simply need to do the job to make sure that they have a roof over their heads and food on their table. Many scientists I know will tell you they feel guilty that they're even getting paid to do what they do because they're having so much fun. And so it's conveying that thrill of discovery and that thrill of being on the cutting edge of a field and that thrill of working with people who are really bright and really talented and really creative. So I would tell anyone before you walk away from a career in science, try to consider some of these things, because maybe there are a lot of other career paths that'll be financially rewarding and maybe even safer. But boy, I just think it's such a privilege to be a scientist.
Yeah, and I think often it's because there's so many failure, like there's sort of more failures and more things that go wrong over the years, and then you get that one or two, really big things, and so it seems unbalanced. But yeah, I definitely agree. We need to promote more of the positives for
Yeah.
sure. And then kind of within that, in particular, if you stay in science, I know I've had this conversation a lot with my trainees of deciding between academia and industry. What is your advice that you give trainees?
Yeah. Well, fortunately, I think you can do great science in academia, and you can also do great science in industry. And I really think it comes down to finding the best cultural fit for you, and also deciding what sort of trade-offs you're willing to make. So I placed a premium on the freedom to work on what I wanted to work on, and with whom I wanted to do the experiments, and I did not want to be in a position where someone was dictating to me what our project was going to be, what our priorities were going to be. Now, the trade-off is, yes, I have to write an occasional grant and seek funding for my work. But on the other hand, if you're in industry, you may be writing progress reports. Some people are attracted to sort of the sort of team science that sometimes happens in industry. But I'm at times sort of a lone hunter-gatherer scientist. I like to follow my nose and follow my curiosity, and I don't want other people telling me what I should do, because I think at this point my scientific instincts are pretty good. And so again, it's just a question of what trade-offs are you willing to make.
Yeah. And I to that other point about more team science, I think, if someone were interested in that, there are a lot of opportunities for that. I think like my career has been a lot of team science. So definitely there's, you kind of have to figure out, which environment works best.
Yeah, and as you know, there's ample opportunities for team science even in academia today. And to me, what's fun about that is often these teams sort of assemble and disassemble based on the challenges at hand. And sometimes increasingly they involve investigators from multiple institutions and multiple parts of the country, and sometimes even multiple parts of the world. So that's the way I like to do team science.
Yeah, maybe I'll do one more question that I had, and then I'll go to an audience question. So I really liked the advice you had kind of earlier on about, your experience with struggling in a lab, and, realizing sometimes it could be your fault, or maybe just the wrong lab or mentor or environment. And, I've come across many trainees who've, struggled in particular due to a lack of inclusiveness in their environment, either their lab or they felt in science in general. And so what are your thoughts on inclusiveness and how, labs or, the scientific community can work on being more inclusive?
Yes. Well, such an important question. And I suspect both of us wish the answer was simple, but I think there are many, many layers to it. I mentioned, first of all, that I think so much of scientific training is done by apprenticeship. And I can only imagine it helps if at least there are some opportunities for mentors who look like you and share some of your cultural experiences. And so we have to do a much better job diversifying the pool of potential mentors. I mean, believe it or not, for me, even the fact that David Livingston was a physician scientist was helpful for me, because physician scientists tend to think about science differently than a PhD scientist. So even having that shared cultural experience for me, having a mentor who understood what it meant when my beeper went off and my patients needed me to go to the emergency room, I mean, that was helpful. So I think we have to diversify the pool of mentors. But now you're going in the other direction. I think every study I've read suggests we're losing talented individuals from, for example, the underrepresented community, really early to science. I mean, in some cases, even before they get to college. So we have to figure out a way to sort of reach in now at the front end to get young people engaged, including people who represent underrepresented minorities. And I think part of what I'm hoping we can do with this new series that Damon Runyon is sponsoring is sort of demystify a little bit the path to becoming a scientist, and at least maybe plant a seed in certain people's minds that, maybe I can do this after all, because I think what they're going to hear from other scientists, similar to the path I laid out, is it wasn't a linear path where everything fell into place. In fact, I think more often than not, we made quite a few mistakes along the way. But at some point, we had that aha moment that I think I can do this. And again, I think mentorship is really an important part of that.
Oh, great. All right, I'll go to some questions from the audience. So thanks for posting your questions. So the first one comes from Benjamin. With the extensive training associated with becoming a physician scientist and leading a successful lab, how did you avoid burnout through your career?
That's a great question. So, as I said, when I was younger, I really wanted to be a clinical doctor. And so I did get a lot of clinical training. Sometimes people will say, well, if you knew you were going to be a physician scientist and a laboratory-based investigator, would you have done all of that clinical training? But first of all, I think you can see that having some clinical training did inform and color the research we later did. So I certainly don't think the time was wasted. Secondly, I have sort of a simple rule, which if at any point in time, you're learning new things, you're surrounded by talented people, and you're getting paid for it, that's a pretty good deal. So, I wouldn't change anything about my clinical training. It was an exhilarating time in my life, and I enjoyed becoming a clinical doctor. But likewise, when I started working at least in David Livingston's lab, and really saw science done at the highest level, I really felt like a kid in the candy shop. And so I think part of the answer for avoiding burnout is it really didn't feel like work to me. It felt like I was playing. And I think, again, that's part of the secret here. I think if the project feels like drudgery and you're just dragging yourself into the laboratory, either it's a terrible project or maybe it isn't, maybe this isn't the right career path for you, but it should feel like fun. I think if you're in a good lab and have a good mentor, it should feel like fun. That helps with the burnout, that it didn't feel like work, it felt like play. And then once I started my own laboratory, I was very mindful that I wasn't willing to compromise in terms of being a good husband and a good father. So I did make sure there were ample time to spend at home and enjoy my personal life with my family. And I didn't allow my scientific life to be all consuming.
Yeah, great, thank you. So another question from Roni. Are there any daily habits you had when you were training that, looking back at now, you think helped set you up for success in science?
Oh, what a great question. Well, let me cheat and first answer a related question. I think I've been struck over the years how many scientists, and I'm talking mostly about biologists, actually did come from worlds like mathematics, physics, computer science, where the premium was on learning how to think clearly and logically, and that never goes out of style, and you can apply that to whatever you do, right? So I think one thing I got right again was I think my mathematics training and computer science training was actually prepared me very well for biology later on, and it didn't hurt me that I took so few biology courses in college. But in terms of daily habits, I tried to be relatively well organized. I mean, all this is simple stuff. I tried to have a reasonable number of objectives for what I wanted to try to accomplish every day. I tried to give some thought before I left the lab each day. Is there something I can do right now that, if I invest 10 more minutes now, it'll actually save me an entire day tomorrow. I'd have little checklists of the things I wanted to do, things I wanted to accomplish. And I think I was pretty careful and pretty conscientious about my work. If people showed me a new technique, I took meticulous notes on how they did what they were doing, because I wasn't smart enough to know yet what things you could sort of cheat on and what things you couldn't cheat on. So I really literally, anything they did, if they coughed, I wrote down they coughed, and so I knew I could reproduce what they were doing. So I think I just basically was careful and conscientious in the lab and very well organized.
Yeah, that's really important, actually, I found. Yeah, awesome. Right, from an anonymous attendee, the US has invested enormous amounts of resources through the NIH into cancer research over the past few decades. Nonetheless, the mainstay of therapy for many cancers has not changed. Is there a problem with the type of science we fund or the way we fund science, or is the problem of treating cancer just that difficult?
Really a great question, very important question. So in my mind, the big bang in cancer research didn't really start till the year 2000, when we had the first draft of the human genome. In fact, it's pretty remarkable what we did figure out about cancer before we had the first draft of the human genome, because to a first approximation, as you know, cancers are the result of mutations in specific genes. And until the year 2000, we didn't have the list of potential genes. And it was only shortly thereafter that we had the technology to rapidly figure out which genes were defective in which cancer. So the analogy I use is, at least starting in the year 2000, we at least had the parts list. Imagine you're trying to fix the car. The first thing you need is the parts list. And then you have to start to figure out what the different parts do. So I think the clock in my mind didn't really start ticking in earnest till the year 2000. And then when you consider that even once you have a well-validated target where you say, if I only had a drug against this target, it would be effective for this cancer. Historically, that has taken as long as 20 years sometimes, depending on the nature of the target. Now, fortunately, we've accelerated that a lot. Sometimes it can even be done in less than 5 years. But I don't think it's that surprising that we're only seeing this wave of precision cancer drugs over the past decade. But then, to be honest, our knowledge of cancer is still incomplete. You can have the parts list, but we're far from knowing what every single part does. And we're far from understanding, well, if this part's defective, what's the therapeutic? Some parts lend themselves to drugs and some don't. So, I also like to point out that the reason Kennedy could be sure it would take 10 years to put a man on the moon is because all the science you needed to know to put a man on the moon was known by 1960. I think cancer is, at this point, partly an engineering problem, but it's still partly a scientific problem where we're still learning some of the rules. So I know it's frustrating. Some of you on the call know I lost my wife to glioblastoma in 2015. So I know it's not happening fast enough, but it is happening faster. And the only thing I can promise is the more we invest in research, the faster the knowledge is going to come and the faster we're going to get rid of this horrible disease.
Thank you for that. So from Paul, how do you decide when to give up on an idea versus trying different assays when experiments fail? This is a great question.
Yeah, it is a great question. I'd be interested in your answer. This is one of those things I assumed I would get better at this over time, and it remains just as hard now as it was when I got started, because there's always the temptation to say, oh, if I only worked on this another month or two, we'd break the back of the problem. But as the question anticipates, this is a really important decision because often young trainees are linked to these experiments. And so maybe at this point, I'm okay if an experiment fails, but my trainees don't have that luxury. So sometimes I do have to say, look, you've been doing all the right experiments. It was a great idea, but you need a gusher. And right now this looks like a dry well. So we better go drill elsewhere.
Yeah, that's a similar answer so far as, the time of a PhD or so. And a question from Marlon. Would you please share one of the first observations you made using the microscope your mother gave you?
We had a pond down the street from my house. So I went down to the pond and just got some water from the pond. And I was looking at all these little microorganisms under the microscope, and I thought that was fascinating. Now they had some pre-prepared slides as well. I remember there was a Drosophila wing or something like that, but it was really cool looking at the pond water. So I think it was the pond water.
Yeah, what's funny is I completely forgot that I also had a microscope when I was really young, like a Fisher-Price one that had like presets. So I don't know, maybe I have a daughter, maybe I'll give her a microscope when she's all right. I don't know, Young, did you want to wrap up maybe? Yeah, I'm getting a message. Yeah, so we're at the time, so I think it's a good time to end, and thank everyone for your questions. Those are really, really great questions, and thank Bill again for taking the time to talk about your path. It was really interesting.
Thank you both for a really inspiring, presentation and conversation. And thanks so much to our audience for all of the fantastic questions and for joining us today. So please watch your email for a link to complete a post-webinar survey and provide feedback and suggestions to our team for future events. And stay tuned for details on upcoming webinars later this year. We're going to be announcing our next webinar, which will take place in October, and we'll focus on using technology to minimize disparity in healthcare and research study design. Thanks again for joining us. Visit our website at damonrunyon.org to sign up for our newsletter and to read the latest exciting news and discoveries from the brilliant young scientists that are being funded by Damon Runyon. So thank you again for a wonderful and fascinating conversation, and we look forward to seeing you all again very soon. Have a great evening or rest of your day.
Thank you all.
Bye-bye. Thank you.
Thank you. Bye.